So now just to summarise the digestion of and absorption of carbohydrates and proteins. There's different sizes of carbs, and you can absorb the monos, you can't absorb the polymers unless you digest them, and there's some polymers that you can't even digest. Dietary fibres come from fruit and veg and cereals, and they can either be soluble or insoluble, and that will determine whether they can be fermented or not by colonic bacteria. And there's different kinds of dietary fibres, so 45 is mostly starch, which contains amylose and amylopectin, amylose being straight chain, amylopectin being branched. Starch is the plant storage, whereas glycogen is the animal storage. You can also have dietary monosaccharides, which you can absorb, and dietary disaccharides such as sucrose, glucose and fructose, lactose, glucose and galactose, and maltose, glucose and glucose. Carbohydrate digestion takes place in two steps, so that's luminal digestion by the amylases, and then membrane digestion by the brush border disaccharidases. The disaccharidases are mostly active in the jejunum, they're less active in the duodenum and ileum, and not in the colon at all. Lactase is one of those brush border disaccaridases, and if you have lactase deficiency, you won't be able to absorb you won't be able to break down the lactose into glucose and galactose, so you'll have diarrhea, cramps, hydrogen in your breath because of the colonic bacteria metabolizing the lactose, and a smaller rise in blood glucose because you're not breaking the lactose down to glucose and galactose. Once you've broken down these, the disaccharidases, the oligosaccharides into the monosaccharides, so glucose, galactose, and fructose, they're then taken across the apical membrane into the enterocyte via two mechanisms: SGLT1 sodium glucose transporter, that's for glucose and galactose, and the GLUT5 transporter for fructose. So fructose, galactose and glucose, as well as fructose, cross the apical membrane via SGLT1 and GLUT5 for the fructose, and then they cross the basal membrane via GLUT2 into the blood. And then looking at proteins, protein absorption and digestion is a bit similar, that's why they're both sort of bunched together, but proteins, you break them down to oligopeptides and then amino acids for enterocyte uptake, but you can also absorb them intact, so that's infants from breast milk, and also immune function from Peyer's patches. Sources of protein, well, 50% in diet and 50% is endogenous. The luminal protein digestion starts in the stomach because of the gastric chief cells producing pepsinogen, which is activated to pepsin. That starts the protein digestion, down to digest 10 to 15% of the proteins. And the luminal protein digestion continues when the pancreatic acinar cells produce zymogens, such as trypsinogen, which is activated to exo or endopeptidases. And the acinar cells are stimulated by cholecystokinin as a result of the fat and the LCRF pathway. Activates the other enzyme to endo or exopeptidases. Exoendopeptidases result in oligopeptides, whereas exopeptidases result in amino acids. The oligopeptides are then dealt with at the brush border by brush border peptidases. So that's shown here, digestion by brush border peptidases, and that's if the oligopeptides are large, but if they're small, then they're taken up by enterocytes via pepT1 enterocyte absorption, where they're digested by cytosolic peptidases. Gives you amino acids, which then crosses the basolateral membrane. I looked at the four routes of protein digestion and absorption, the fact that they can either be digested to amino acids in the lumen, digested to amino acids at the brush border, or oligopeptides can be taken up directly into the enterocytes, where they can be digested to amino acids, or they can be absorbed into the blood. There's four transporters, there's four mechanisms by which the apical amino acids are taken into the enterocyte, and that will vary depending on whether they're cationic and cysteine, neutral, anionic, or glycine and proline. And once they've been imported, then you need to efflux them, and the efflux will depend on whether they're cationic or neutral. So if they're cationic, is this pathway, neutral is this pathway, and 10% of the amino acids that are imported into the enterocyte, they're used by the enterocyte for protein synthesis. What can go wrong? Well, some diseases can inhibit the amino acid import into the enterocyte, as Hartnupp's disease and cystinuria. They result in a defect in the import, but there's no protein deficiency because you can still absorb oligopeptides. And as shown here, Hartnupp's disease inhibits the B0 neutral amino acid cotransporter, whereas cystinuria inhibits the B0 plus cationic transporter. What does cystinuria result in? Well, it results in cysteine in the urine, giving you kidney stones. Hartnupp's, however, results in dermatitis and lack of coordination, brain ataxia, because you're not absorbing those neutral amino acids. And then finally, look at the fact that some proteins can be absorbed directly, so in their intact form via two routes for an immune response, either phagocytosis by enterocytes or phagocytosis by M cells in Peyer's patches, the lymph patches, where the antigens are essentially transferred to lymphocytes for an immune response.